just to present now the kind of disorders that we're working on at the Florey Institute. And I'll mention a few of them today, but I'm sure you're all absolutely familiar. And I can imagine if you think about yourselves, if you think about your close relatives and close friends and go through this list, I would be very surprised if even a single one of you could not think of a close relative or yourself or a close friend who is not afflicted by at least one of these brain and mind disorders. So I'm not going to go through and name them all, but the reality is, in terms of total burden of disease, that brain and mind disorders, including all neurological and psychiatric disorders, dwarf the rest of the burden of disease outside of the brain and nervous system. And we don't necessarily get the funding we need for these major neurological and psychiatric disorders. So it's an ongoing unmet need that desperately needs more funding going forward. Here's another way of looking at it, and I'll present different stages today. Obviously, there's an emphasis on brain aging, but I'll tie in the idea of brain development and brain health across the lifespan. And if you arrange it this way, you can see these various disorders of brain and mind and various neurological and psychiatric disorders arrayed across the lifespan. So there's effectively no stage in the human lifespan when we're safe from one of these brain disorders or disorders of the nervous system. And all of these disorders listed here are being studied at the Florey. We're trying to understand disease mechanisms, and by doing that, trying to understand ways to prevent, to treat, and ultimately to cure this range of terrible disorders. Just looking at this burden of disease and specifically focusing on brain and mind disorders, they affect around 1.5 billion. And as I mentioned, this is an increasing burden of disease around the world. 25% of the total burden of disease, and that's from the World Health Organization. And we know that 75% of the population will suffer from a brain or mind condition in their lifespan. And we know even this year there's been such a focus, understandably, on COVID-19, but increasingly we're realizing that the COVID-19 pandemic is likely to be followed by a mental health pandemic. And I'll mention later on some of our research which ties in directly to infection, including infection with the SARS-CoV-2 virus. Here's our general approach. We know that every disorder, including brain and mind disorders, results from a complex combination of genetics and environment. And that's one of the key things that we're trying to understand here. I'll mention a little on a particular so-called genetic disorder, Huntington's disease, where we were the first in the world to show that, in fact, Huntington's can be affected by the environment, not just genetics. I'll touch upon schizophrenia and autism, but particularly focus on Alzheimer's disease and other forms of dementia and disorders of brain aging, but also the concept of brain health throughout the lifespan. I'll talk about enviromes. That refers to your total environmental exposures throughout your lifespan, and some of the environmental factors that we're interested in, including cognitive stimulation, physical activity, stress, diet, and sleep. I'll be talking about the brain, obviously, and the Florey's world-leading expertise in studying the brain, both in humans, but also in important preclinical models of these various disorders. So the brain is unique in a whole range of different ways. And one of the aspects where it's really unique is that it never really stops developing. What you're seeing here in this image is that you've got this brain here over a period of time, and it's a human brain, and these images are being generated by human brain imaging, an area where the Florey Institute is very strong. And you're going across this timeline from 5 years of age through to 20 years of age, nominally adult, although interestingly, the brain may still be maturing at the age of 20. And what this is showing down the bottom, as you see these colors in the brain on the left, is looking at the human brain from above, or the colors are obviously artificial, but they're based on neuroscientific data. And over here, you're looking at the brain from the side, actually, if you were looking from the right side of an individual. And what you're seeing with the colors changing over time is over this period of 5 to 20 years of age, what you're seeing is a sculpting of this important cerebral cortex that encases the outside of the brain. And this thinning or sculpting is driven by the environment, by experience. So the brain is continually responsive to environmental factors, to experience. And this is a visualization of the human brain being sculpted by the environment, by experience, from the years of 5 to 20 years of age. And this is just one of the ways in which you can really see the excitement of neuroscience and the kind of things that we're doing at the Florey. I'm going to be talking today about 3 distinct revolutions that are occurring in science and where our Florey Institute research is heavily involved. And these revolutions will be around neuroplasticity, around genomics, and epigenetics. And so to introduce this, we need to think about the human brain and the fact that it's not a hardwired computer, but it's an extraordinarily complex organ, perhaps the most complex known object in the known universe. However, everything that we think, everything that we feel, and everything that we do is generated by this 1.5 kilograms of soft tissue, grey and white matter in the skull. Once again, these color coding of the different functional regions of the brain. And importantly, we think there's around 100 billion neurons in the brain. And these are interconnected by approximately 1,000 trillion synapses. So extraordinary complexity, and these are the challenges that we're really dealing with to understand the healthy brain in order to prevent, to treat, to cure a whole range of different brain disorders. Okay, to introduce this, I first used this slide 4 years ago, and it happened to be just after the US election, and you'll see the context here. So starting with the concept of Franz Joseph Gall, and back in this period in the late 18th and early 19th century, we knew almost nothing about the brain. And so Gall used to feel the bumps on people's heads and infer their brain structure and therefore function from that. And this phrenology was later discredited as a science. However, we can think about the type of what phrenologists used to say, and this was more relevant 4 years ago when Hillary was running for office, a profound thinker versus a shallow thinker. And we've all lived through the last 4 years, and we're now past a second election, and I'll discuss that no further. All right, so to move on to this first revolution of neuroplasticity. And it's only recently, in recent decades, that we really understood that the brain is so highly plastic. It was originally thought to be, once you reached adulthood, to be more or less fixed, like a hardwired supercomputer. But we know that's not the case. And this plasticity in response to environment and response to experience continues throughout your life, including middle age and later stages of life. These are the kind of technologies going from that brain imaging that's based on a technology called magnetic resonance imaging, which I'll talk a little bit about later. But again, the Florey is very strong in this area of brain imaging. And these images are when you just use a technology that just shows the nerve fibers or the axons coming up through the spinal cord and coursing throughout the brain, including the cerebral cortex. And this is one way of looking at the structure and the function of the brain. And we know with these kind of techniques that and other techniques in preclinical models that learning occurs through plasticity. And this involves changes in the strength and number of connections, and can even be the number of cells. You can generate new cells in adulthood later in life. And ultimately, we need to think about the fact that neurons that fire together, wire together, and environment and experience makes them fire together and changes the structure and the function of the brain, not just during development, but throughout life. We also know in particular studies that changes in the volume of brain tissue can occur with specific forms of learning. And this has been seen in musicians, taxi drivers, students learning a language, and even something like juggling. So to introduce this, I mentioned magnetic resonance imaging, or MRI, one of the great strengths of the Florey Institute. And you can see here, this very expensive MRI machine where the subject will lie here with their head here, and then be inserted into this very powerful fluctuating magnet, which will allow the imaging, not just of the structure of the brain, but also functional changes and blood flow changes and so on in response to neural activity, which can be used to study structure and function in the healthy brain, but also how structure and function goes wrong in a whole variety of brain disorders. So using this technique, these investigators who I knew from my days in Oxford, they did a really interesting study where they took volunteers, these happened to be Oxford undergraduates, and they allocated them into two groups. These are undergraduates who'd never actually juggled before. And they randomized them into a group that learnt juggling and were taught to juggle to an expert level, and those who weren't, the control group. And the key thing to understand here, using this brain imaging, and the red areas mark areas that changed after learning to juggle, that they could see, even with this structural technique, that learning to juggle led to structural changes in these nerve fibers and axons in the human brain, a dramatic demonstration of what we call experience-dependent neuroplasticity within the human brain. Okay, I'm moving on now to the second revolution, which is a revolution of genetics and genomics. And so we're living in very exciting times whereby just 2 decades ago, around the year 2000, when the first human genome sequence was generated from just a handful of humans, and that cost billions of dollars to sequence a single genome at that stage. And now 2 decades later, a single human genome, which is 3 billion paired letters of DNA, can be sequenced very rapidly for less than $1,000. Some claim a few $100s perhaps now. And the challenge is we can sequence it rapidly, and I think ultimately this will lead to precision medicine and personalized medicine, so that when you go to your GP or another healthcare professional or specialist, you might actually have your genome on a credit card type device, or they might download it from the cloud data from a centralized server, for example, obviously protecting genomic privacy and so on. And that will be the starting point for diagnosis and so on. But there's such enormous complexity here that it's really just a start. We really don't know how a genome helps generate a human from a fertilized egg, and how it affects whether you get a disease or you don't get a disease, and so on. One of the areas we've been particularly interested in is repetitive DNA, or stutters, in the human genome. So I coined the term repeatome, which refers to approximately half of the human genome, which consists of repetitive DNA, repeating letters of DNA like a stutter that occur throughout the human genome. And it turns out that this repeatome, and particularly these tandem repeats in the human genome, are really important in a whole range of diseases, including a disease that I'll show you some examples from, which is Huntington's disease. And so we've been working on this important human disease for a couple of decades now and made a whole range of important contributions internationally to our understanding of what causes this disease, and also potential new therapies, and some of our discoveries in this preclinical model of Huntington's disease with our clinical collaborators are already being translated for new approaches to prevent and treat Huntington's disease. Just to introduce Huntington's disease, one of the more famous individuals to suffer from Huntington's disease was the great American folk singer, Woody Guthrie. And in fact, we've written a review article recently because we were the first in the world to show in a preclinical model that we could model the depression that occurs in approximately 50% of these people with the Huntington's gene mutation, and help understand that important symptom, not only within Huntington's, but more generally in the population. So the title of our articles is Why Woody Got the Blues. And you can see in the written text there, I'm not going to read it all out, but the crux of it, as he developed this movement disorder, these writhing dance-like movements, that's why it's called Huntington's chorea, that he was ridiculed as being drunk, and people thought he was a drunk. And so on top of having this nasty fatal disease, people from Huntington's families were accused of being drunkards and discriminated against at every turn. So just one of the more horrible diseases that can be afflicted upon anyone. And one of my favorite musicians and singer-songwriters of all time is Bob Dylan. And he visited his mentor and hero, Woody Guthrie, and Woody was on his deathbed as Huntington slowly killed him. And Bob Dylan wrote this song. And this song could have been written in 2020. Hey, hey, Woody Guthrie, I wrote you a song about a funny old world that's coming along. Sick and it's hungry, it's tired and it's torn. It looks like it's dying and it's hardly been born. That was Song for Woody by Bob Dylan. All right, so focusing on the disease now, I mentioned it's a fatal brain disorder caused by this genetic stutter, this expansion of a repeating tract of DNA letters. One of the nastiest things in this disorder is that if a person has the disease and has children, their children are 50% likely to inherit this fatal and very nasty disease. It's a complex combination of cognitive problems that culminate in a form of dementia, of psychiatric symptoms, the most common of which is depression, and the movement disorder, this chorea, this writhing dance-like movements that I mentioned before. Unlike some other disorders of neurodegeneration and brain aging, this often hits in midlife, often in the 30s or 40s, but in 5% of cases, it's juvenile and childhood onset as young as 2 years of age. And in those cases, it kills these children within usually a few years. Here's these repeatome or these tandem repeats. So Huntington's is the most common of a range of different disorders, most of which affect the brain and other parts of the nervous system. And they're all a different genetic stutter in a different gene in the human genome. So things that we discover within Huntington's have insights and potentially relate to new therapies across a whole range of other disorders of brain and mind, a whole range of other neurological and psychiatric disorders. And this is an area we're pursuing as well. And in fact, one of the repeats actually is the most common cause of both ALS, which is the most common form of motor neuron disease, but also FTD, which refers to frontotemporal dementia. So this work has major implications, not just for Huntington's disease, but for motor neuron disease, for other forms of dementia, and so on. And also Fragile X syndrome, these disorders of brain development as well. This is again just showing the human brain side on, where you can see here in blue the cerebral cortex lining the outside of the brain. It's very important for our thoughts and our mood and various other functions, our behavior. It's a key area affected in Huntington's, and this structure called the basal ganglia, the blue bits in the middle, are also heavily affected. And in a brain that's affected by Huntington's, you can visibly see when they donated their brain after death, whole areas of the brain that had basically disappeared from this patient who slowly died of Huntington's over 10 to 20 years. All right, so those of you interested in genetics might enjoy this slide. So those affected by Huntington's disease are generally heterozygotes. So essentially, they have the stutter, the gene mutation, on one copy of one gene from one parent, and usually the other parent didn't have the disease, so they had a normal copy of the gene. And so understanding the genetics of Huntington's is an important starting point, whereby we launch into a whole range of other studies. So this is one of the key studies that we did, just really summarized here, whereby we worked with the key preclinical model of Huntington's. In this case, it was transgenic mice that expressed the human gene mutation. And so they developed memory and cognitive problems. They developed behavioral changes which are relevant to depression in humans. And they developed movement problems which model chorea in Huntington's disease. So extremely accurate model of the disease. And I can tell you the various discoveries we've made in the last 2 decades using this mouse model of Huntington's disease are now being trialed in the clinic by our clinical colleagues in Melbourne and elsewhere. So the key to this, which relates not just to Huntington's, but relates very much to Alzheimer's disease, to Parkinson's disease, and other disorders of brain aging and, in fact, brain development. So you can see here that the key difference here with environmental enrichment, it's modeling increased cognitive stimulation and increased physical activity by increasing the level of novelty and complexity within the environment. You can see the kind of brain regions that are stimulated. And this was the study actually performed with my first PhD student. And essentially what we discovered was that when we divided these mice into either environmentally enriched, where they had increased cognitive stimulation, physical activity, versus standard housed, the environmentally enriched Huntington's mice with more cognitive stimulation, physical activity, had a dramatic delay in onset of disease. This is the first demonstration in any genetic model of a human disorder that environmental stimulation and exposure could be therapeutic, and has led to a whole range of research, not just in my group at the Florey Institute, but by many colleagues around the world. So one of the things we try and model are the cognitive deficits modeling dementia in Huntington's disease. And we can't get them to solve these crosswords, for example, but we can get them to do really complex tasks that sometimes involve touch screens like iPad type devices, and they get a strawberry milk reward when they get it right, and so on. And these type of tests directly translate to the clinic. And that's been a focus of our work in recent years at the Florey Institute as well. We're also trying to understand the psychiatric symptoms such as depression that are so prevalent, not just in Huntington's, but a whole range of other disorders, including obviously major depression itself, which is one of the most common and devastating disorders amongst the population. And so when you're using a mouse as a preclinical model of these disorders, it's not easy to model something like depression, but fortunately we do have ways of understanding depression in mice, including taking an antidepressant drug such as Prozac or Zoloft. And when we give it to the mice, we can change their behavior, and we can change it in a way that makes us confident that the data that we get is highly relevant to clinical Huntington's disease. And we can do exercise interventions in the mice and show beneficial effects, antidepressant effects, which relate to the beneficial effects of exercise in humans with respect to antidepressant effects and elevation of mood. And so taking this environmental enrichment paradigm, we want to explore specifically the role of exercise, which is obviously beneficial for the body, but we know it's beneficial for the brain as well. And this is one of the major areas we've been pursuing at the Florey Institute in recent years. Can we understand the beneficial effects of exercise? I've coined the term environmemetics, which includes exercise memetics. These are novel therapeutics which mimic or enhance the beneficial effects of cognitive stimulation and physical activity. So if we can understand how cognitive stimulation and physical activity have therapeutic effects on the brain, we can identify new therapies, including drug therapies, which can act perhaps synergistically with cognitive stimulation, physical activity to prevent, to treat, and eventually cure a whole range of different human brain disorders. So here's some of this complexity. And one of the things that has kept me in neuroscience all these decades and keeps me passionate is not only the fascinating aspects of the brain, but its beauty. And so you can see here just these single neurons within the cerebral cortex of the brain have such beauty and complexity. Each one of these single neurons can receive over 10,000 synaptic inputs. And so there's such complexity and such beauty within the brain. And if we can understand it down to the level of cells and molecules, we can ultimately prevent, treat, or cure any brain disorder known to man. And that's the overarching goal of our work at the Florey Institute. Here's an example. When you zoom in, this is a single neuron making synapses or connections with each other. And these synapses are particularly important, we think, and they are the type of things that go wrong in disorders like Alzheimer's disease and other forms of dementia, and something we need to understand right down to the level of cells and molecules. Here's another example of neurons. These happen to be from another part of the brain called the cerebellum. And when you use these neuroscience techniques to study them, you can see this exquisite complexity and beauty. And again, a single neuron, in this case, a Purkinje neuron of the cerebellum, can have approximately 200,000 connections or synapses with other neurons. Just mind-blowing complexity that we need to grapple with in order to find ways to combat a whole range of neurological and psychiatric disorders. And then using very powerful microscopes, we can zoom down onto particular processes of neurons and look at these individual synapses and understand how these synapses are affected not only by disease, but by environment. This environmental enrichment, increased cognitive stimulation, physical activity, changes the structure of the brain as well as its function, including the addition of new synapses onto existing neurons. So exercise, we know, can lead to release of important brain growth factors. It can cause generation of new neurons in the adult brain, something we call neurogenesis. It can enhance learning and memory and therefore act as a cognitive enhancer. It can increase blood flow to the brain and other beneficial effects in the body, and therefore be beneficial in terms of preventing disorders like Alzheimer's disease and dementia, for example. And we're increasingly looking at not just studying the brain in isolation, but the brain embedded in the body, how the brain continually interacts in a dynamic way with the body. It's complex bidirectional interactions, to the extent that we know now that, and I've been a co-author on a recent article in an international journal, that even something like COVID-19 affects the brain and nervous system. There's a neurological and psychiatric component, we think, to COVID-19 infection, which reflects the fact that the infection starts in the body, but by infecting the body, these viruses like SARS-CoV-2 can affect the brain and nervous system. And so that's a level of complexity we need to approach, even as we understand the effects of infection and pandemics on the brain and nervous system, and hence on neurological and psychiatric disorders. Okay, the final revolution I'm talking about is the revolution of epigenetics. And so I think this has been covered recently. There's an ABC series called Superhuman, and they've interviewed me and various other people for this. And I think on that, I provide some analogies of epigenetics. I'll repeat this again. There's two analogies that might help you understand epigenetics. If you like to think about text and reading, think about every cell in your body being an instruction book for the body and the brain. And essentially think about the 20,000 or so genes as being 20,000 or so words in that book, every cell in your body. And so think about epigenetics. If the letters of DNA are the letters of those words, then epigenetics are things like underline and bold and italics and even fluorescent highlighters that dictate the extent to which each of these 20,000 genes is turned on and off in different cells of your body at different stages of life and in different disease states as well. If you prefer a musical analogy, then think of the 20,000 or so genes as being 20,000 or so musical instruments. And so you then think about epigenetics as being the musician. So when you bring the DNA or the genes that are the instruments together with the musicians being the epigenetics, together you produce the symphony of life. This is a more technical way of showing these beautiful strands of DNA and these chemical modifications and molecular changes to DNA that are epigenetics and control expression of these genes in different cells at different life stages and also in different disease conditions. And so things like environmental factors, stress, diet, behavior, toxins can have epigenetic effects, which have long-term effects that can switch individual genes on and off. And so just showing them epigenetics as being gene switches that can be controlled in every cell of your body. This is another way of think about this bell curve of brains. Each of these brains has been generated by a different genome. And at the edges of the bell curve, you see the red and blue brains. We can think about as brains associated with people who have a particular disorder. In this case, think about them as disorders like schizophrenia and autism spectrum disorder. So evolution occurs slowly. So we effectively have the genomes and the bodies and the brains of our caveman hunter-gatherer ancestors. So if any human disorder is changing within decades or a couple of generations, it's not a change in genetics. It can't happen that quickly. It must be a change in environment and a change in exposures. And that change across the population, and this may again relate to COVID-19 and pandemics, that can change the shape of this bell curve so that the brain phenotype, even though the average brain phenotype may not have changed, the edges of the bell curve are altered. And so you may get increased brain disorders at these edges, these red and blue brains. And we've put forward this idea that that's why certain brain disorders, like autism spectrum disorder, may have increased in recent decades in the last couple of generations. And we're actively exploring that at the Florey Institute. In fact, we published a very exciting paper this year with Melbourne colleagues where we provided the first evidence that in male mice before conception, that infection of those males before conception changed the epigenetics of their sperm in a way that changed the phenotype of the offspring. This has enormous implications. And at the moment, I'm seeking extra research funding for my group and put forward a proposal where we will explore this in terms of COVID-19, both effects via fathers, but also effects via mothers. The idea that someone infected with and coming down with COVID-19, or maybe even infected with SARS-CoV-2, the virus, the coronavirus, and not showing symptoms, this may affect their sperm, their eggs, their in utero environment, and therefore affect the offspring. One possibility, it could lead to increased disorders, including brain disorders in the next generation. So this is something we've described in a recent publication, are actively pursuing. We just desperately need more funding to be able to do these key experiments. This is a broader idea, the idea of these exposures in males before conception, including stress, including these environmental factors like physical activity, also including diet, change the epigenetics of the sperm, and then at fertilization with the mother's eggs. This can then lead to changes in brain development, maturation function, which could lead to changes in things like cognition, mood, and behavior, which would make someone potentially more prone to a particular brain disorder. And we're interested in the environmental exposures on the maternal side with the mothers as well. So I mentioned diet, and this article came out of studies in which the diet of males before conception changed not only the epigenetics of sperm, but change the offspring. And this may be relevant to being overweight or obese and metabolic syndrome. And we think things like overweight and obesity and many other disorders, including perhaps cardiac disease, perhaps diabetes, could be primarily disorders of the brain. Because if you think about these so-called lifestyle disorders, the biggest problem is addiction to sugar, addiction to food, addiction to other substances, problems with behavioral control where you overeat or eat inappropriate substances. And so those lifestyle factors, which are driven by the brain, can lead to increased things like diabetes, to cardiovascular disease, to cancer. And so one perspective on this is that the vast majority of common human disorders have their origins in the brain, in cognition, in behavior. And that's what we primarily need to understand. Getting back to this concept of brain aging, thinking about this concept of use it or lose it, so that there's neuroprotective brain capacity due to chronic enhancement of cognitive and physical activity. And at the Florey Institute, we're studying how environmental enrichment and cognitive stimulation, physical activity can be therapeutic at the level of genes, at the level of epigenetics and molecules, at the level of cells, and at the level of whole brain systems, right through to cognition, to mood and behavior. By doing this, we think we can develop new therapies that ultimately will prevent, will treat, will cure a whole range of different brain disorders. This is the overarching schema of my group's work at the Florey Institute. I used to say we're all dealt a genetic deck of cards at conception. It seems with our new work and the work of our colleagues that we're also dealt an epigenetic deck of cards at conception. For some of us, with these blue arrows, because of our genetics and epigenetics, we start to head down the pathway of brain dysfunction, which predisposes us to particular brain disorders. These positive environmental factors like increased cognitive and physical activity may build up a brain reserve that's protective. Someone who already has a dysfunctional brain may still be able to undergo functional compensation and brain reserve via positive factors like cognitive stimulation, physical activity, reducing stress, a healthy diet, and so on. And even someone who develops a brain disorder, we're using this approach to develop novel therapies that I call environmemetics, a subset of which are exercise mimetics, that could push people with a brain disorder over to this side where we all want to be, which is healthy brain function and aging. So it's an enormous and complex puzzle. Our work at the Florey is trying to understand the mechanisms of these various disorders and ultimately use this information to develop ways to prevent, to treat, and to eventually cure this range of devastating neurological and psychiatric disorders. So for you, you wonderful people at U3A and your friends and family, what's a general message that comes out of the neuroscience? These are things we can say now based on public health. And you, this audience, I'm sure already knows it, that things that are good for your body are also good for your brain. So staying physical activity, physically active, even just walking, just low intensity physical activity is good for the body and good for the brain. Being a member of U3A helps keep you mentally active and cognitively stimulated. So I think your approach to lifelong learning, brain training is going to help delay onset of things like Alzheimer's disease and other dementia. Obviously, eating a healthy diet is important for everyone. Managing stress levels, and this may relate to the other environmental factors, is very important. Stress is bad for the brain, but it's also bad for the body. And maintaining healthy sleep patterns, which in the era of where everyone's surrounded by screens and digital technology is become harder than ever. And so sleep is important, not just for children, it's important throughout your whole life. So finally, and this slide was again 4 years ago, and Sean Connery has unfortunately left us, one of the great actors, but Sean, I recall, made it into his 90s, and part of it was keeping physically active, no doubt cognitively stimulated as well. A healthy diet and minimizing stress where you can do so. And the endpoint of the US election is certainly going to reduce stress for some people around the world. Okay, so you can see here, but seriously, I think this year and this COVID-19 pandemic, we're worried about an ongoing pandemic of mental health, the stress that it has caused and the changes to lifestyle. We need to understand what's happening so that we can anticipate perhaps increased brain disorders and ultimately prevent them or at least treat them. So I'd just like to finally acknowledge these absolutely wonderful people at the Florey, they're a great inspiration. Throughout this year, throughout this pandemic, these people have worked so hard. They're often on low salaries, staff and students. They work phenomenal hours. You wouldn't believe some people are working over 100 hours a week. You know, they're so dedicated, they're so passionate and so undervalued. And I thank them and I thank you. Thank you very much.
Thank you so much, Tony. That was truly fascinating. I'm sure everyone who's tuned in today has learned lots of new things, and hopefully will take away their learnings and share them with their friends and colleagues and family. We've had a few great questions come through, which obviously show that you've sparked a lot of curiosity from our U3A community. So hopefully we'll get round to half a dozen. If there are any questions that have been sent through that we don't have time for, we apologise, and we're happy to answer them via email. So our email, as mentioned, is the fundraising@Florey email. So let's get to the first question, which is from one of our special supporters, Tony. Arturo has asked some questions about Alzheimer's. He's curious to know, despite the fact that Alzheimer's has been around for hundreds of years, and that it has a massive economic and emotional cost to society, which is constantly rising, and despite the funding which has been generously provided by donors and governments, why is there still no cure for the disease? So where are we at, I guess, with Alzheimer's today at the Florey?
Okay, look, the bottom line is not enough funding. Governments don't fund us enough. Unfortunately, we can't raise enough funding from other sources. I think with more funding, we would have cured all of these diseases. I believe that. I believe in science. I believe in medical research. But you ask another important question embedded in that, which is why these disorders are so common. And the reality is, I mentioned we've got the genomes and bodies and brains of our hunter-gatherer ancestors. And so selection acted on when people were reproducing in their teenage years and 20s. And so most people died of other disorders before they got old enough for Alzheimer's and dementia. So natural selection didn't act on those particular genetic factors. And so, you know, by being a modern society, we're kind of running in the face of many aspects of evolution. And so, but we do need to understand evolution in order to develop this precision medicine and personalized medicine, which is what we're trying to do at the Florey.
Thanks, Tony. We've got a question now from someone who is anonymous, and they're talking about what you mentioned, exercise, looking at Sean Connery, for example. So the question is, how long lasting are the effects on the neurons and synapses induced by exercise or cognitive stimulation?
Look, they're great questions. And I think from a public health perspective, you know, epigenetics can lead to long-term changes. And so the reality is, you know, someone who is physically active in early life and midlife may gain benefits in later life. But for all of these environmental factors, the greatest benefit is if you maintain it throughout life. So not to do something and then stop it. So if you can maintain physical activity, continue it. If you can maintain a healthy diet, continue it. If you're cognitively stimulated, try and continue that throughout your life. And same with reducing stress and sleeping well. They can have long-term effects, but the greatest benefit is maintaining a healthy lifestyle throughout the lifespan.
Mm-hmm. Consistency, the key, right? Like your research. A question about your particular research, Tony. Have you studied the effects on OCD in your research?
That's a really interesting question. We've got an incredibly talented PhD student with one of my colleagues we're starting to co-supervising. He is working on obsessive compulsive disorder, very complex. It sits under the spectrum of anxiety disorder, and we do other work on other anxiety disorders, extremely complex, difficult to understand. But in the next few years, OCD is one of the psychiatric disorders, the brain disorders that we're going to be working on. Not well understood at all.
Okay. We might just wrap up with two more questions, I think. This one's quite an interesting one. Thanks, whoever shared it is very personal. They've expressed that their father survived the Nazi concentration camps and is wondering about the links between their own anxiety and depression and their father's experience.
Look, that's a great question, and you have my sympathy. And I can tell you, in terms of the human data relating to that preclinical data I showed you, one of the key studies that's been published and that informs our work are Holocaust survivors. One of our international colleagues has studied multiple generations of Holocaust survivors, and there's indications that there are transgenerational effects going through multiple generations that can't be explained by anything except epigenetics, that it must be epigenetics. And so other forms of trauma as well, I think globally the stress and trauma of COVID-19 this year could have impacts across the next generation, maybe multiple generation. Other people have studied 9/11, the attacks in New York back a couple of decades ago and finding similar things. And so that the human studies support the idea that this great stress and trauma can have transgenerational effects via epigenetics, and that's part of what we're trying to understand.
Okay, and I think the last one to wrap up on is also very pertinent to the year. It's in terms of stress, but Erica from Wangaratta is curious to know about the optimal level of stress. So obviously there's a balance. How does that work, Tony? The good stress versus the bad stress?
Look, the thing I love about you 3A is they're such intelligent questions, such highly informed questions. And it's absolutely true. You know, we evolved with stress, and our stress system evolved to deal with stress. So, you know, no one can have an absolutely stress-free life. So it's about how you control and manage stress. And the greatest psychological stress is unmanageable stress, where you can't do anything about it. And your stress levels are high day to day. Having just a busy lifestyle where you've got deadlines and a lot to do is not a bad thing. Like the low level stressors that you deal with and you handle and you're on top of is not bad. It's just the unmanageable high levels of stress that are chronic, that start to affect the brain and body.